Hello. Hello, how are you? How's everyone doing?
I have stuff all over my furniture. I've acquired a cat.
Oh, yeah.
You acquired a cat.
Yes.
Out in the country, you got how that the cat walked more than 15 minutes.
She's been hanging around here freezing to death for quite a while.
Okay.
And so she was really, truly freezing to death. And Jim picked her up and brought her in. And we now have a barn cat in the house.
You can eat your barn instead.
Anyway, hi everyone.
Hello. Hello.
Okay, Bradley, I just got your paper. I'll get to it sometime later, and we'll try to merge and see what happens.
Yeah, it sounds great.
Okay.
I mean, it looks good. Yeah.
Your version yet.
Oh yeah.
Good.
But yeah, we'll merge them and then try to submit it somewhere. And I think it looks. I mean, like it's pretty good.
Well, I'll try to submit it back where it was invited, even though it's late.
Well, yeah.
See what happens.
I think they probably, special issues, they take what they can get, I think, a lot of times.
Maybe.
We'll yeah.
Okay.
But
I'm
yeah,
not worried about it.
Yeah, I know. So, Susan, how's your, you presented on the tensegrity stuff last week. How was that?
Oh, yeah. They took off the interesting final part about the cells and embryos, but I think it went fairly well. I had someone very interested in it because he was trying to use hyperelastic material and for a J curve, and then I said, well, tensegrity did it, and he was floored. He's asking a whole pile of questions. So apparently, J curves of any sort works better to model tissue. Go figure.
Oh yeah, well.
Yeah.
Maybe we'd fall into a puddle on the floor.
Yeah.
Anyways, I'm having fun with my modeling. I upgraded my program and it undid a kind of complicated 3-cell structure, so I get to redo it.
Ah.
Yeah.
Redo what?
Your
the presentation?
Pardon?
You redo the presentation?
No, just my model.
Oh, okay.
Yeah. It's annoying.
Yeah.
Yeah. I would like the newer version of console.
Yeah. So it's hard to keep up with the latest versions on that.
Well, the latest version will automatically make a stress strain map without me having to go to all kinds of gymnastics to get one.
Wow. That's great.
Yep. So I want the newest version. I told them that. Whether I get anywhere or not is another question.
Great. So, Sushmanth and Gautham, how are you?
I'm Bradly. How are you?
Hello?
Can you hear me?
Yeah, I can hear you.
Cool. This weekend I took a break. Haven't worked so much. It's like I was mentally caught up with so much work and just took a break for 4 days.
Yeah.
I went a trip. That's all with me, and I was about to keep a PR by today night. That one in Huggingface to create a complete interface of DevoLearn. And I was thinking to upload DevoGraph also because it all belongs to Devo only, right? So I was thinking to host our models there. Maybe I was thinking to keep that spaces also, like whatever DevoLearn has spaces right now. I want to create a 3D space. I mean, a space which generates the 4D structure of a graph.
Yeah, that would be good. I was looking at it yesterday.
Okay. Actually, there is a thing we need to run after every 3 days, we just need to create a check. I mean, we need to run these. If we don't run them, they will just stop and they won't work. From now on onwards, I will take care of that. Due to that, we need to create a run after every 3 days. For freer version of Huggingface, we need to run that whole space after every 3 days.
Huh.
Maybe I will integrate with some code, or I will just run manually
Yeah.
to run those spaces.
Okay. Yeah.
I want to complete. I will completely host this DevoLearn Huggingface. Like just we had spaces right now. I want to create a complete DevoLearn models will be hosted, like cell membrane, nuclear segmenter, lineage population model. With that DevoGraph also, and a README about our DevoLearn will be hosted. I will create an API and I will share you with that by today night.
That sounds good.
And Gautham also did pretty work. Like last meeting, Dick has mentioned about centroids, right?
Right.
To keep a different color. He actually did that whole work. We discussed about it now only just few minutes back.
Oh wow.
Gautham, you can share and show the.
Yeah.
Yeah, sure.
Okay, so like we discussed last time, I just made some few changes to the code, and now we have the cell membrane and the centroid in different color. And the small changes, I just added a new animation function so that we can just compare it with the raw footage and see how our model is performing.
Okay.
So to get this two different colors, I had to make some small changes in the DevoLearn report, because till now what we are doing is we are just passing the video to the DevoLearn, and they used to give us the output as it is. So we couldn't change anything about it in the DevoGraph code. So I made the changes to DevoLearn report itself, and I'll just clean the code a bit and keep the PR by tomorrow.
That sounds great. So the one color is the membrane segmentation, or the edges of it. The other is the green, which is the centroid.
Okay.
Yeah.
Remind me, please, did this data set include the nuclei?
No, it didn't. It's just this green mark is the centroid. It doesn't look like there are any markers in there.
Okay, if you get a data set that has the nuclei, then a third color would be appropriate.
Yeah. Yeah.
Okay, then we can see.
We need to know the truth of the data set actually. That's what I don't know, like they have tried with the original one or not. I have gone through those sets also. I didn't find one. I was still searching other data sets to put them.
Yeah. Well, I think the EPIC data set has like nuclear stain. Whether that really marks the nucleus very accurately or not, I don't know. But yeah.
Yeah, we'll go through it and we'll work on that.
That sounds great. Yeah, this looks pretty good.
Yeah, thank you. Other than this, I haven't worked much this past week.
Oh no, this is great. Yeah. So yeah, I look forward to that pull request. And thank you, Sushmanth, for your work on the Huggingface. I was looking through the Huggingface setup, like the account yesterday. It looks like there are elements of GitHub. Does it interface directly with the organizational GitHub repository, or is it just using the elements of GitHub? Because it looks like it's using like a standard repository structure from GitHub. And
they are actually using standard repos from GitHub.
Okay.
I mean, we can't integrate completely because actually what all the LFS files, actually whatever our repos are there, they contain large file systems. Actually, the repos already occupied the all LFS free GitHub resources. So I can't integrate Huggingface and this DevoLearn completely. That's the main issue. If anyone wants to contribute in Huggingface, they can just do keep a PR for that also,
Yeah.
like we do in GitHub. And I was thinking to completely create a same interface like GitHub and Huggingface. Whoever want to contribute in Huggingface, they can contribute there, and it will be used for Huggingface community, and we can showcase there. And GitHub will be also. I mean, I need to do changes in DevoLearn README file also. I will do complete PR. I will show you a small presentation of time thing in next. Maybe by night I will work on it. I will keep a PR for that, or I will just create the whole thing, and I will let you know.
All right. Well, that sounds great. Yeah, I look forward to that pull request as well. Looks like it's coming along really nicely.
Okay. When can we submit our draft for GSOC? I mean, first draft. Can we start now from now on?
Well, yeah, the application period starts, I think, coming up soon, but like you can send it to me anytime if you want me to look at it. I'll probably want to look at it at least once, you know.
Okay.
So the application usually is like where you state the problem, and that's just basically restating what what I've put up there as the call for projects. And then you would talk about like what you're trying to do, what your approach is, and then you go over a little bit of, you might put some images in. A lot of successful applications have had several images, embedded where they show kind of a demonstration of what they're doing. It doesn't have to be like really involved because you're just starting out. And then you kind of finish up and say what you want to do, give a schedule. If you go to the GSOC website, they have the dates for the program. You can kind of put it in terms of the dates that you want to, like targets that you want to hit during the program, during the coding period. So the first 2 weeks of the program is community building, and that's not really expected to be too much work on your end, but you do have to, get everything ready to go. And then the coding period, you have, you want to hit milestones every week. So you want to be able to get, like week 1, you want to be able to make sure that you have everything running, no problems. And then, week 2, you want to start with a small task. I've had people in past years divide their work up into 2 or 3 sections. So they start with like one, that you've already been working on getting models into Huggingface. You might follow up on that during the coding period, or you might have like some other thing that you're doing during the coding period. And, the idea is that you don't want to, it's a pretty quick pace. So you don't want to fall behind. You want to have a reasonable flow of activities, but you also don't want to say that you're going to do more than you can do. And that's sometimes a problem because people do that and then they get in trouble, they fall behind. So, then like you should always build in a little bit of time to catch up. At the end, maybe 1 or 2 weeks at the end where you can finish up things. And then that should even out. So, things won't go exactly as planned, but at least you have a good sense of the timing and the rhythm of the project. In past years, I've had people who have run into problems from, based on what they've proposed. So the proposal will say, I'm going to do these 3 things and I'm going to get through them in a certain amount of time. And then that'll be the project. And then they hit a stumbling block where some plugin doesn't work or, something isn't working the way they want it. So that's a problem that you have to think about when you plan out your project. If I can't do this, or if I'm running late on this and things are going slowly, then what else can I pick up on during that week so I don't fall behind? So you have to think about those things when you write your proposal. So then once you write your proposal, that's pretty much it. You want to put your CV at the end, your resume, and then that's the whole proposal. That should be probably, 5 to 8 pages. I think that's what they want. I don't know what the limits are. They change it every year, but that's pretty much what I'm looking for. They can help you with stylistic things, or if I think that something is too ambitious or too broad, then I'll tell you. Because I've had that in the past where people have proposed things that were really broad, and it's like, well, that seems like a lot to do in 1 week, or that needs a little bit
Bradley,
more
what's
specificity.
your deadline? What's the deadline?
I think they submit in April, I believe.
Okay.
So we have time. So yeah. And then
we'll just submit our first draft by next week or next 10 days. I mean, we need to see a lot of things, so we'll try to submit our first next 10 days.
Well, it doesn't. Yeah, at this point, it doesn't need to be in a very finished state. Just sketch it out, and I can tell you whether you're on the right track. So, I mean, I just want to make sure that you don't go way off track.
Okay.
Yeah. So,
that's all my phone. Yeah.
Okay. Well, yeah, thank you, both of you, for the update. That's great. Looks like Susan has a interesting paper I ran across, and this is inhomogeneous mechanotransduction defines the spatial pattern of apoptosis-induced compensatory proliferation. Well, that looks interesting. Yeah. So this is, let me see if I can share my screen here. I just pulled it up. So yeah, this is an interesting looking paper. So this is a lot of words, Susan, in the title. Why did you find this interesting?
Because it shows how apoptosis rearranges cells.
Okay, yeah.
Yeah, there's some shorter titles that come up when you look for that. I can't get this paper yet because it's too new, because it's February 2023.
Right.
But let's see. I've got mapping the dynamics of force transduction at cell-to-cell junctions that came up. That's an older paper. And then cellular mechanotransduction from tension to function. So there were a couple that just sort of came up when searching for that. So that's partly why it's interesting. It's giving me some other papers.
Oh yeah. Well, I mean, it looks interesting. I always find it funny when people like write a really technical title and then, sometimes it scares people off. But this looks like.
I don't know why they had to have that title.
Yeah.
How about apoptosis creates new patterns in cells? Anyway.
Yeah. So the highlights, only a restricted number of neighboring cells are selected to divide upon apoptosis. So, usually apoptosis is this programmed cell death. So it's usually something in development, you get programmed cell death, in a, like you'll have a lineage and it'll expand. And then at some point, this, some cells will die off depending on their spatial position or maybe like, due to crowding. If you think about the hand in, or the forelimb in tetrapods, which are mammals and a couple other groups, they have this sort of web. We start out with a web. It's sort of our hand with their fingers, and it sort of starts out by default as a sort of a webbed structure. I'll allow you to show my Jamboard here. I'm drawing on it. So it starts out kind of as a web structure like this, actually more like this. And then the cells in between the digits undergo apoptosis. And this is just a made-up limb. This area here is the area that dies off. So the cells in this area undergo apoptosis, and then you end up with these digits that then go on to develop the fingers or the digits of the forelimb. So this could be like a paw or a hand, or any sort of number of structures. And it in evolution it changes slightly so that the pattern of apoptosis changes a little bit. So it sculpts this tissue. It sculpts these interstitial areas here. And so it's just a matter of these cells dying off in the right pattern. Are you going to say, Dick?
If you don't have it, you end up with a duck.
Yeah, webbed hands or webbed feet or whatever.
Yeah,
it works in the feet as well.
It's actually a rare condition in people.
Yeah.
Mhm.
Yeah. So that's what they mean by apoptosis, and so only a restricted number of neighboring cells are selected to divide. So this is another one of these sculpting mechanisms where you get cells that either proliferate or die off. The pattern of division is defined by inhomogeneous YAP-mediated mechanotransduction. So that means that the mechanotransduction is where you have a cell, and that cell is subjected to forces or mechanical forces, and those mechanical forces act sort of as so you have your cell, you have mechanical forces coming in, and it senses the mechanical forces, and the cell behaves accordingly. So we know that mechanical forces, when it's experienced pushing or pulling on the cell body, it can be, can have different responses. There are transcriptional cascades that occur because of this. So that the forces, as information is transmitted to the genome and things are, transcribed here. And so we get this sort of, I'm not showing this part of the screen. Okay, so this is it. This is my drawing of the mechanotransduction in the cell. And so you have these forces that act upon the cell and changes its behavior in different ways. And what they mean by inhomogeneity means it's not homogeneous, it's heterogeneous. And so that leads to the third point. Inhomogeneity arises from non-uniform force and nuclear size in neighboring cells. And so we know that non-uniformity is sort of the key to some of these processes. So this is a graphical abstract here. And this will have a little bit more, probably better in my drawings. So this is where mechanotransduction occurs. This is the cytoplasm, the nucleus. This YAP factor is activated during mechanotransduction. So mechanotransduction happens in cells with a large nucleus and a large mechanical stimulus. And so this is an example of apoptosis, where the cell dies in the middle, this one cell here. Then this is where you have spatially inhomogeneous mechanotransduction, and this cell in blue as opposed to the red. So the red cell is apoptosis, then the blue cell is this inhomogeneous response. And then in this white cell down here, this is a small nucleus and small mechanical stimuli. So it is not it doesn't experience that mechanotransductional whatever it is that they're proposing here. And then that means that this blue cell will divide and fill the space formerly occupied by the red cell that are known apoptosis. And so it fills in the cells that die off. Those spaces get filled in by the cells, sort of selectively speaking, get filled in by these cells that are experiencing mechanotransduction. So basically, you're selecting for cells with a large nucleus and large mechanical stimulus, and it's filling in eventually all these spaces. So it's not like the example of going from the webbed forelimb to the two digits. It's that the entire mass it remains a mass, but it's getting filled in with cells with a certain competency, as opposed to the ones that die off in apoptosis, or the ones that eventually, maybe even the ones that don't have this competency for mechanotransduction or YAP transmittal. So this is YAP is the yes-associated protein, which means, I guess, that it's accepted by the nucleus. So these acronyms are not like mysterious. They're just kind of like made up. Like they just make them up on the spot. It's the yes-associated protein.
So my favorite is Sonic Hedgehog.
Oh yeah.
Yeah, really? Why did they name it that way?
Apparently because people like the cartoon, and they just gave it a name. They gave it the name. It's a weird story about gene naming. Like people would name them things just randomly, and then like they do gene therapy where they have to talk to people whose children have diseases, and they have to talk about the Sonic Hedgehog protein. And there's been this debate about whether they should say, well, maybe we should change the names to something more serious. I don't know. Anyways, that's good, Susan. That's a nice paper. I don't know if we can get access to the full paper, but I think that basically tells us what we need to know. Okay. Yeah, so the DevoLearn repository, it looks good. I had a couple, of course, we've had a couple of pull requests accepted. We have, I think, a number of issues that have been closed, but we have new issues being generated here. Thank you, Sushmanth and Gautham, for doing work on this. So this is DevoLearn, and of course, if you're watching this and applying to Google Summer of Code, we have a lot of issues that if you go to the repository and you look through it, you may want to create an issue, which you're welcome to do if you think that something needs to be improved. If you think that maybe you want to work on something, this is another place to put it in this issues. If you want to propose a change, you can issue a pull request, and you're welcome to do that for both DevoLearn and for DevoGraph. And so DevoGraph is the newer version of this. It sits on top of DevoLearn, and it has the graph neural network capabilities, or at least that's where we're headed with this. We're headed towards this. We're building out this pipeline. So again, start an issue or issue a pull request, and that's where we're going to be working from this summer. So.
Yeah, okay, I'll check out the issues.
Yeah. I think the issues are.
Some issues need to be closed because, like, I think I need to close them. I have worked on 2 issues. They just need to be closed. Actually, I will mention about this issue and I will close them.
Yeah. It'd be
There
good.
are not 8. There are 6 issues to be closed.
Okay.
And in DevoGraph also 1 issue was been closed. Whatever issue was there, I think my idea of approach was wrong, so I have closed that issue.
Okay.
But in DevoLearn also, we need to see this. We need to close some issues. Bradley, did you archive that repo actually? I mean, what we call DevoLearn web? Yeah. Sorry.
Yeah, I did archive that, and we actually have some new public repositories, and these are the ones that are the models on Huggingface, so nucleus segmenter, membrane segmenter, and lineage population. These are a little bit more specialized. These are for DevoLearn, so these plug into DevoLearn, and this is the home of the stuff that's being put up on Huggingface. So we're trying to do like make these available in this community, which is a lot of people do machine learning, and you can put the model in here and run it. So we have this, and I guess we'll be updating this throughout the summer with different. I guess this will be something that'll be worked on over the summer. Is this correct, Sushmanth? Or
hello?
Yeah.
Okay, I will just I didn't understand what you're telling. Actually, my
Oh,
net was like,
is this something that is going to be like worked on over the summer? Is this something that someone could put an issue in if they think that there's something they want to add to it, or how do we want to
yeah.
do this?
I mean, actually, I want to integrate both them, like from GitHub and what we call. A minute. I mean, GitHub and Huggingface will be integrated. Everyone, when someone gives a pull request, it will be directly pushed in Huggingface. Also, I want to create that workflow for these models. And for DevoGraph also, I was thinking to create something like we need to deploy in Huggingface and show people, like create a 4D structure of the graph.
Yeah. So that would be just a separate thing here. So this is the DevoWorm group. Yeah, I've made an organization card. I don't know why it's not coming up, but and then this is the Huggingface. I guess they have like teams or organizations or whatever this is.
Okay,
And then
I will create this whole interface, Bradley. I think I haven't worked this week. If I worked out, I would have completed this whole thing. By today night, I will keep a pull request for those three also. I will create a model card, and for the I will host the models, and I will upload the data sets what we have actually.
Okay.
I just refracted them. I will just upload there, and we can show everyone.
Yeah.
Same like GitHub interface will be there in Huggingface, so everyone can easily integrate and see it.
Yeah. Okay. That sounds great. Thank you. Yeah, that's great. So something I found interesting, I know Dick last week mentioned about Thomas Kuhn and incommensurability. So I found this, or I was looking into it afterwards, and found this. This is a philosophical blog, I think, the Multidisciplinarian, and they talked about this sort of Thomas Kuhn's work and some of the work he did on this idea. So Thomas Kuhn, of course, wrote the book The Structure of Scientific Revolutions, and that was in 1962. Of course, that was very influential. A lot of people learn about that book. They learn like that book, and
Bradley, he was surprised at that. I read his book a few years after the first edition.
Oh, wow.
I wrote to him about it, and he said he was surprised that anybody was still interested.
Yeah. Yeah.
Anyway, okay, go ahead.
Yeah. So it's usually you either learn about this book or like Popper's book when you learn about philosophy of science, if you're just doing science. Like if you take a philosophy of science course, there's a lot more to it, but those are the two books that people usually talk about. So structure is where Kuhn proposes this idea of a paradigm shift, which is where if you have a theory or if you have a field of science, you have a set of theories, and sometimes there is a paradigm shift in which some idea or finding will overturn previous theories and create a new theoretical paradigm. So that means a paradigm shift. And so, as Kuhn saw it, the scope of a paradigm was universal. It's not merely a theory, but a framework and worldview in which a theory dwells. And so it's, in some fields, and it's debatable as to whether that shift is like a revolution or it's more like just you shift to a different theory that, because I guess it depends on the field of science. So, a lot of theories will be revolutionary, but they'll build upon previous ideas. So, like Darwin's theory of evolution built upon things that were going on before Darwin, in terms of understanding the natural world and understanding the role of fossils and in the history of life and things like that. But the theory of evolution itself, or the idea of natural selection, was revolutionary. So, but anyways, this is the sort of the philosophy that he's come up, that he's most famous for, is this idea of a paradigm shift. And so he explained that successive transition from one paradigm to another via revolution is the usual developmental pattern of mature science. And by developmental pattern, he just means like the history of ideas, not like developmental biology. His view is that paradigms guide research through periods of normal science, during which any experimental results non-consistent with the paradigm are deemed erratic and discarded. And so, there are examples of that throughout the history of science. It's just basically what are people finding the most relevant in any one time. I mean, this persists until overwhelming evidence against the paradigm results in its collapse and a paradigm shift occurs. And then he stresses this idea of incommensurability between associated paradigms, meaning that it is impossible to understand the new paradigm from within the conceptual framework of its predecessor. So this is like the Copernican revolution, plate tectonics, or quantum mechanics. So if you tried to explain the concepts, or you tried to you tried to use the measures of quantum mechanics using classical mechanics, you couldn't really explain it. You need a new framework. So if you have like certain measures and you say, I'm measuring something at the quantum scale with quantum measurements, it isn't consistent with classical mechanics. So it would be very confusing to sort of integrate those measures into a classical mechanics model. And so you have to come up with a new model for it. And so that's exactly what happened. The Copernican revolution is the same thing. So as people were measuring the positions of the planets, they came to the conclusion that the geocentric universe wasn't a sufficient model. So you have these conceptual models of how the world works or how the universe works, and you have your sort of measurement techniques and your measurement, actual measurements that you're making, and sometimes they are inconsistent or incompatible. So that's sort of the idea, I guess, behind incommensurability, where there's this mismatch between the sort of the framework or the mental model and the actual measurement work that's being done.
Yeah, Bradley?
Yeah.
Before you go on, I've written a comment about this, and that is, in biology, this doesn't seem to happen.
Oh.
And that no paradigm ever dies.
That's interesting, yeah.
Yeah, biologists seem to be immune to changing ideas.
Yeah.
So part of the problem is Kuhn only dealt with the physical sciences. He never really got into biology, except in this posthumous book.
Oh, okay.
And there he got into it in ways that you probably would appreciate. But he never examined biology itself as a discipline.
Oh, wow. Okay. Yeah.
Okay. So I think that was a flaw of his, but it was a limitation. He was raised in the physical sciences.
Right. Yeah, it's always, philosophy of science. So it's like, what science are you talking about? Or different patterns in different sciences, and
yeah. So I've noticed this. It's almost impossible to develop a new paradigm in biology and have it accepted universally.
Yeah.
Maybe that's why the biomedical engineers are getting in the act.
Oh, yeah.
Maybe.
Yeah, the fellow that was using hyperelastic J curves to model cells was floored that I'd used tensegrity. He said, "What? There's a J curve there?"
Well, this actually it has been going on for a long time. The whole development of molecular biology, if you look at its history, came from physicists around 1950. Okay, it required people from outside the field to get into it, to end up introducing a new way of thinking, and then it got stuck.
Yeah.
I find biology as more of an observational type of discipline. They just say the cell did this, and that. Plunk.
Yeah.
Well, yeah. But if you take people like, Crick was a physicist. The whole revolution of thinking about DNA came from physicists. Oh, there were a bunch of the other ones. A bunch of the pioneers of molecular biology were physicists. Okay, and now they're sort of at the periphery in terms of molecular dynamics and stuff like and engineering and stuff like that. But I can't say you can't say they're now central to biology yet.
Well, they're coming up with some interesting theories.
Oh yes,
Yeah.
oh yes. It's a question of who gets hired eventually.
Yeah. Well, it's also a question of the different departments that house people. Like, you have molecular biology, and a lot of times it's very distinct from like integrative biology, and
Oh yes,
yeah,
it's
so
the fragmentation. The
yeah,
fragmentation of science. Yes.
right.
Yeah, well, there used to be, well, I could have easily been a biomedical engineer right from the get-go, but there was no such thing when I graduated from engineering. There's
Yeah.
no
Yeah.
such thing.
Hey, you're right. When I got into computers, there's no such thing as computer science.
Yeah.
Oh, okay. Well, there you go.
We all show our age.
Yeah, well, I took computer programming in grade 12 as a spare extra frill course.
Oh, wow.
And I had to do it with punch cards.
Yeah, well,
Yeah, and they didn't have a computer on site. It all had to go to the university to be run through.
oh yes.
Yeah.
When I started out, we had punch cards and 24-hour turnaround to a computer from Oregon going to California.
Okay, yeah.
So,
Yes, it's just very new, really.
Yeah. So yeah, the article they talks about so it talks a little bit about incommensurability, and talks about so I guess this person is going to focus mainly on his aspects of work and popular conceptions on it related to its appropriation in technology and business process management. So this is kind of their point of view. I'm not going to go into that part, but his claim of incommensurability, that's Kuhn, led many to conclude that the selection of a governing theory is fundamentally irrational, a product of consensus or politics rather than objective criteria. This fueled flames already raging in criticism of science in postmodernist, subjectivist, and post-structuralist circles. Kuhn was an overnight sensation and placed on a pedestal by all sorts of relativism, sociology, arts, and humanities movements, despite his vigorous rejection of them, their methods, their theories, or paradigms. Then decades later, the Rhodesian structure, which I guess is the follow-up, I guess that may be the posthumous book that he.
no, it isn't.
Oh, it isn't? Okay. But this would be later on after the 60s. Kuhn added that if it was relativism, it was an interesting sort of relativism that needed to be thought of before the tag was applied. And then communities outside of hard science, 20th century social theory in particular, couldn't get enough of Kuhn and his paradigm shifts. So it was very popular in terms of the sort of the idea, but whether that actually represents what's going on in a lot of sciences, I mean, that's, it was more popular to think about it that way, because sometimes ideas are popular because very consistent with what we want to think of as the way things work, instead of like actually how the way things work. I guess that was the point he's making here. So some models are more aesthetically pleasing than other models, and the less aesthetically pleasing models might be more right than the other ones. But that seems to be, and of course I think there are philosophers of science who talk about that. So that's actually quite different than the paradigm shift, in the sense that you get these things. It's like, okay, how do you change the way we look at the world? Well, sometimes it has to be aesthetically pleasing more than actually consistent with the data. So we have some things in the chat.
I put the new book in the chat.
Oh, okay. The last writings of Thomas H. Kuhn. Yeah. Okay. I have to look at that. That's from this year or last year.
Yeah.
All right. Yeah. Thanks. Sushmanth had to leave. He's having network issues. He'll give an update in Slack. Thank you, Sushmanth. Yeah. And so this was, yeah. Okay, yeah, I think that's good. That's a nice article. And yeah, I'm glad we were able to follow up on that. I think that's an interesting concept and interesting set of ideas. And yeah, it's definitely something that we should consider when we think about some things we talk about in the meetings. And yeah. So actually, I wanted to talk about this paper as it just came out. And I know our people left, people who are most interested in this, but they can catch up later on this. This was posted in the Slack the other day. I found it this week sometime, and it's a new paper. It's brand new, and it's on using these graph neural networks in C. elegans. So one of the things about our project on graph neural networks was that we didn't really have a good example of it in C. elegans, and it was kind of like one of these things we just had to imagine how it would be useful in C. elegans and building these, embeddings that would, maybe be good for building networks of cells. But this is actually on adult C. elegans, in fact, aging C. elegans. And this paper is a GNN-based model, which is graph neural networks, for capturing spatiotemporal changes in locomotion behaviors. So this is where they're looking at behaving adult C. elegans. They're looking at their movement behavior. They're tracking this, movement in a culture dish or something like that. They're tracking it, and then they're using that tracking data to build these graph structures, and then they're using graph neural networks to analyze that. So the abstract reads, investigating the locomotion of aging C. elegans is an important way for understanding the basic mechanisms behind age-related changes in organisms. However, the locomotion of aging C. elegans is often quantified using insufficient physical variables, which make it challenging to capture essential dynamics. So, sometimes with locomotion, we'll use tracking the motion using just kind of like visualizing the worm in some way. We need to have better physical variables for that. So this makes it easy. This makes it hard to sort of understand the movement dynamics. Sometimes, for example, you might use some fluorescent marker. You can put fluorescent markers in C. elegans in the head cells, so you can see the head is a little green dot moving around. Or in the tail, you might stain some neurons, so you can see this trail of green going through the field of view. So there are different ways you can do that, but that's not the best way to do it. So, so to study changes in the locomotion pattern of aging C. elegans, we developed a novel data-driven model based on graph neural networks, in which the C. elegans body is modeled as a long chain with interactions within and between adjacent segments. So they're building a model of the C. elegans body. They're taking the data from movement and they're integrating it with this model. And so the C. elegans body is modeled as a long chain. Their interactions between different segments of that chain, and then their interactions are described by high-dimensional variables, which then can be reduced using some sort of principal component analysis or some sort of dimensionality reduction. Using this model, we discovered that each segment of the C. elegans body generally tends to maintain its locomotion. In other words, it tries to keep the bending angle unchanged and expects to change the locomotion of adjacent segments. So it tries to not bend its body that much, but it actually also expects that the adjacent segments will change their locomotion. So it's sort of like when they undulate back and forth, they're trying to maybe keep stability going forward. So they're just kind of moving their body back and forth. It's kind of like when you drive a car and you try to keep going down a straight line down the road, you move your hands back and forth on the wheel slightly just to keep the car in the lane and going straight. So it's like this, positive feedback, or, you want to be able to go over maybe different types of terrain and maintain stability. The ability to maintain its locomotion strengthens with age. So as they get older, they're able to maintain their locomotion better. Besides a subtle distinction, I guess, in the changes in the locomotor pattern of C. elegans at various aging stages were observed. So they were able to observe changes in this locomotory pattern as the worm ages. The worm, goes through these larval stages, 4 larval stages, and then they become adults. And then they get older. And as they get older, they sort of, even as an adult, they change different aspects of their morphology. But the adult is generally considered to be sort of like, where they stop their development and they're laying eggs and things like that. Our model is anticipated to provide a data-driven method for quantifying the changes in the locomotion and for mining the underlying causes of these changes. So I'm looking for some figures here, and probably at the end, they have a mathematical model in there. Here's a figure 1. So this is the optimization of the model. You have these different segments. This is the worm. You have dorsal contraction, dorsal relaxation. So what's in ventral relaxation, ventral contraction. So you can see that on the dorsal side of the worm, you have these contraction and relaxation segments, or these lateral line segments. And then on the ventral side, you have these, I guess that's the muscle. And then on the other side, you have contrasting. So it's like where you have on one part, here 4, 5, and 6 dorsal contraction. On the ventral side, it's relaxation. So it's like contracting on one side and then relaxing on the other. Dorsally, it's contracting. Ventrally, it's relaxing. So ventral is the bottom part of the worm. Dorsal is the top part of the worm. And so this is where we have this this sort of action, because it works down this. So this is where the worm is undulating, and you have contraction, relaxation in different phases. So as you have contraction here, it's bending in. Relaxation, it's bending out.
Okay. Bradley?
Yeah.
Could you please put the reference for that in the chat?
Yeah. So this is computers. I'll just put the DOI here.
By the way, did you want my presentation? I think it was given publicly, so I can send everybody it.
Yeah. I think that would work.
I'll send it to you and Dick then, or?
Yeah, go ahead. Yeah, do that.
On tensegrity. Okay, sure. I was going to ask, but I realized, well, it's public, so.
Right.
Yeah.
So
did you want the one with the developmental biology on the end of it, or did you want the more?
Oh no, the developmental biology on the end, please.
Yes.
We don't have a problem with that.
That's version 1. He didn't like it. Wanted me to take that out. He says, "Show your own work."
Oh well,
well, my own work crashes, so. And then I got the gears over it crashing from the other professor saying, "Well, you need to see all the matrices to see why it's crashing." I'm going, I don't know.
Yeah.
I don't know what to do with it.
Okay. Bradley, on the top of this paper, I did some work, in the 60s.
Yeah.
Not terribly competent, but I was looking at the question of why grass can grow straight.
Right.
And if you look at grass, it consists of columns of cells which are dividing.
Right.
And I figured there's got to be some sort of a major feedback mechanism, because every time a cell divides, it seems that the whole grass leaf will swing one way or the other.
Right.
Okay? And I actually took some grass leaves and looked at them under a microscope, and indeed they looked like they were swinging back and forth. Okay, so it could be that plants which grow straight, any kind of grass, trees, etc., or anything that's really thin and long, might have some sort of feedback mechanisms keeping it straight.
Right.
Okay? And
Yeah.
that would be similar to the worm, which is keeping itself in an undulatory shape.
Right. Yeah.
Okay? So as far as I know, this is not explored at all.
Yeah, I know.
How would that go for columnar cells in?
Yeah,
Yeah.
well, columns, multiple columns bound together, which is the structure of grass.
Right.
Okay,
Yeah,
or
well, I've got to model that. Polymer cells in a tissue. So I'm not sure precisely where to put the actin ring right about now.
Yeah.
yeah, but I think there's a mechanics of plant growth that is evading us so far, and this may be a key to it.
So would there be mechanics of cell growth or
No,
as
plant.
well? Well, I know
I'm
plant
thinking
growth.
about plant growth in particular. If you take any plant, any grass, take your wheat for example, it grows in straight stalks. How come? How does it do it?
Because it has cell walls?
No, it'll probably end up being falling on the ground, and you would be able to harvest your wheat.
It does that sometimes. If it gets
They do
too
have-
much fertilizer, it'll do that.
Oh, okay. They do have silica bodies, which presumably make them stiff, but I'm not sure that makes them straight.
Yeah.
Okay? Nobody, as far as I know, has ever looked into the mechanics of silica bodies, let alone the overall question of what keeps them straight.
Yeah.
Yeah, that's a good question.
Okay?
Yeah.
So, at any rate, it might be worth some, I don't know, maybe looking into the current literature on growth of grass and see, maybe do a computer simulation of it, see if it requires feedback of some sort to keep it straight.
Yeah. Yeah. In this paper, they use graph neural networks on these different. So they take the worm and they make 23 segments, and then each of these segments is like a part of the graph, so they can measure the different aspects of movement. So
yeah, well, see, the worm has an aspect ratio of maybe 20 to 1.
Yeah.
Something like that. Grass will probably do 1,000 to 1.
Wow. Yeah. So it's this very long chain of.
Yeah.
Yeah.
Okay, and why it's straight is nobody knows.
Yeah. It's basically bending angle at different points. So it's like if you have a stick and you bend it at the top, it bends, it curves down, it bends.
Oh yeah, that's
right.
called Eulerian bending. Yeah.
Okay, Eulerian bending.
Well, it'll bend due to sunlight.
Right.
If it
Yeah.
has sunlight on one side and not on the other, it'll bend.
Oh, but it'll turn. Yeah, that's like turning of sunflower heads. But where does the turning occur, in the stalk or in the flower?
Well, if they get mature, they don't turn. But
Yeah, I know. But if it's we have that. People grow fields of sunflowers, maybe will follow the sun.
Yeah.
Okay. But I thought the turning mechanism there was in the base of the flower. Okay. I'm not absolutely sure about that.
Yeah. So
well, it's more to investigate.
yeah.
Okay. They turn you into a botanist there.
People have been trying to turn me into a botanist forever.
So the
That
thing,
and the nurse, but anyway.
oh yeah. So the way they model this is they model this for looking at bending angles at different time points. So this undulation is over time; it kind of moves, and then these bending angles are measured either for the past m time points, which is this yellow step, or the red and the blue,
So
which are different steps.
this is basically a set of localized control over bending.
Yeah, and then you represent it as this graph where you have these feedback mechanisms at each segment, and so the bending angle can act on each segment, and then it pushes the rest of the worm.
Okay, will the model work if the bending angle is 0? If the goal is 0.
Right. I'm not sure. Probably it would probably be almost no undulation at that point. But
That
then
would be-
probably the
that might
worm
be a-
wouldn't be moving. Yeah.
That might be a place to start on grass.
Yeah.
Well, the undulation here, the reason why they can crawl across the surface, of course, is partly due to friction,
Right. Yeah.
where it touches the surface.
Okay, well,
There'll be friction.
Yeah. So I think the bending angle is sort of like the summary of like the forces against the surface. So like actually in the OpenWorm project or in the OpenWorm Foundation, there's the Cybernetic project, which is cyber, which is with an S, and they have modeled like worms crawling across different media. So like they've got it crawling in different types of gels, for example, and they're simulating different substrate conditions. So the worm will crawl against like a very thick gel or a very loose gel, and the movement is actually different, and the mechanical feedback is different in different conditions.
yeah, there was a very interesting report done in the early 60s where a fellow made a very long trough and put a zebrafish in. The zebrafish had to swim from one end to the other.
Yeah.
Then what he did is he put a layer of milk at the bottom.
Yeah.
And he could see what the fish did as it swam is that it made little whirlpools. And then it pushed against the whirlpools.
Oh yeah, that octopus do that, apparently.
Really?
They create a vortex.
And then push against it? Yeah,
Yeah.
geese may do it too in their bee flying.
Yes,
Yeah.
apparently they do.
Okay, but they're pushing against the vortex created by the one in front of them.
Great.
Yeah, so the lead goose has more work to do than the ones following.
Right, which is probably why they changed leads.
Yes, yeah.
Yeah.
Okay.
Yeah.
Anyway, I don't know if I can dig that up again. It was published as a Navy report.
Oh, okay. Yeah. Might not find that one.
Because they were trying to understand how do you make a boat with less friction.
Oh, okay. Yeah.
I could maybe find the one on the octopus. I was kind of intrigued.
Yeah. So this is a map of bending angle and measured and predicted using their model. This is where you have these loss values for the model. And this just kind of very technical in terms of the model performance. This is sensitivity of the model where you see bending angle measured for different center points and time points. So you can see that there. They actually have HC elegans here for different mutants. So you have like different mutations, and these mutations, UNC25, EGL3, UNC7, UNC9, those are all movement related phenotype. So the mutations are such that they affect the way the worm moves and how they respond to sensory input. So the wild type is going to give you that kind of undulation that's, normal, as they say. And some of these mutants, defined mutants, of course, will give you a different pattern of movement. Will have trouble moving in that way. So that's something they're looking here. And they can see that for some of these mutants, the loss is more severe for their model, not surprisingly. So that's something that they've done as well. I don't know really if they get into some of these network embeddings, which is what I'm looking for here. They're just showing more of the sort of the forward and backward movement connection data here. So these are changes in the connection variables with age. So they have these connection variables, which are these different segments here. It's basically the segments and how they're connected. So they show some of these changes. So in the front, you have more changes than at the back, I believe. And that's true for all these different examples here, D1 through D15. And so you can see that the mean for that is across the adult stages. So this is D1 and D15, which are the adult stages of the worm. And there's variability with respect to age. So D11, it's a little bit above the mean for some reason, and then it reverts back in D13 and D15. Not really sure why that is, but I'm not really familiar with the adult stages of aging, but yeah, I don't know if there are specific things going on. And I mean, later in adulthood, there's variability because they're getting old and they're maybe not moving as much. They've laid their eggs at this point, so so yeah, they're able to model it for these different stages, and they show these different, so these are the different parts of the graph. So they're connecting the segments together, and they're showing differences with respect to age. Yeah, and so that's all they have. But I think that's quite a bit. I think it's interesting work. Yeah, you could probably apply this to plants or other organisms, and it would probably give you some pretty interesting insights. All right. Yeah. So anything else we wanted to talk about before we end?
Yeah.
Okay.
No. I'll send you my PowerPoint. It's just a half an hour lecture. It went well as far as I could tell. People were interested in not going to sleep, so.
Even if they go to sleep, sometimes that means they're still interested. They just have to go to sleep for some reason. I don't know why.
Yeah, I got a reputation when I was a graduate student for sleeping through a lecture and then asking a question.
Yeah.
Yeah. I can imagine the question.
Yeah.
Oh well.
Yeah. All right. Now I'd like to talk about another paper that I wanted to present for a long time, and it's called Universality Out of Order. And it's the paper here. So this is by Petterholm, who's a network scientist. And this is kind of goes over universality and the idea of universal statistical laws. And he's kind of contrasting this with orders, rankings, and hierarchies. And so he's going to go through a number of experiments here and going through some things that might give us some indicators as to the idea of universal statistical laws in everyday data that we see in networks. So orders, rankings, and hierarchies on one side, universal statistical laws on the other. It is where these core concepts of complex system science meet. This comment sets the scene for some of these discoveries in the seldom visited border zone. So he gives examples of pecking orders. So the idea of hens pecking on each other in a fight that allow for dominance hierarchies to emerge, or other types of pecking orders, like who's subservient to whom in a population. It could range from humans to other, like to chickens, to other animals, social groups. And so this is this is something that you'll deal with in data and statistical summaries of networks, where you have pecking orders and social rankings. rankings are then simplified representations of hierarchies. So if we think about like in an organizational structure in business or in government or in the military, you have these hierarchies that are strict hierarchies, meaning that there's, definitely rungs on the ladder, or that some set of individuals reports to another set of individuals, and that culminates in a leader, a singular leader that, is at the top of the ranking. So this is a hierarchy. Basically, we can use rankings to derive a hierarchy. Hierarchy is one of those concepts whose imprecision seems to beget its popularity. There are many more or less problem-specific definitions of hierarchy. These are furthermore often inconsistent. To most authors, hierarchies are collections of levels from top to bottom. Members of the hierarchy would inherit their ranking of their levels, so that one could order members of different levels, but not necessarily members of the same level. So Herbert Simon, who has talked a lot about hierarchical organization in his work, points out the following relationship between hierarchy and ranking. The world is a large matrix of interactions, in which most of the entries are very close to zero, and in which, by ordering those entries according to their orders of magnitude, a distinct hierarchical structure can be discerned. So that's an interesting definition. It's definitely a quantitative definition, something we can work with in terms of building a network structure. And so hierarchies were the basis for Simon's theory of complex systems, which continues to influence us to this day. So a word about hierarchies, and I'll kind of give a little bit of a tour here on this. So this is a hierarchy. We think about this in terms of a binary tree, and of course we use these in development a lot with respect to lineage trees and sometimes phylogenies, where you get this hierarchy of things from the top down to you get it from the mother node to the daughter nodes at the bottom. And so you can follow this hierarchical path downward. There are things that are, so the daughter nodes are subservient to the mother nodes, the child nodes being the daughter nodes down here. So for example, in a lineage tree, the origin point, or the cells up in this part of the tree, provide things to the cells down here. They provide instructions, they provide other types of cues for differentiation, and you end up with differentiated cells in a lineage. But this is more or less the structure of a strict hierarchy, where everything is subservient to something else, where it's contingent on something else. I guess in biology, it's a little bit difficult to use the human analogy. But this is also true in like organizational charts look like this and so forth. There is this competing idea of heterarchy. And we've talked about this. We have a paper, Origin of the Embryo, which was with Rob Stone and Thomas Porteges and George Michalowski, as well as myself. And in this paper, we introduced this idea of a heterarchy, where you have a network, but it's very different from this hierarchical thing. You have this set of nodes that are all interconnected. The interesting thing about this interconnection is that it could be sort of hierarchical in the sense that this node, say for example, can control what the rest of these nodes are doing. These nodes can be subservient to this node. So these three nodes out here could be subservient to this node for a short time. And then control can switch in the network so that this node over here is actually the one that's in charge, I guess, if you want to put it that way. And then these other three nodes are all subservient to that node. And so you can switch off like that over time. And so in the context of this tree, it's really not easy to show the context because it's a fundamentally different structure. And it would be like if this node over here at the in the middle of the tree controlled both what was going on in the top node and what was going on in the bottom node. It's in biology, it's a little bit, in an organizational chart, it would be like a middle manager would dictate what the CEO does and what the base worker does, the entry-level worker. But in a biological network, this, of course, this kind of tree structure is usually unfolds over time. So it'd be a little bit harder for things to be, for the daughter cells to control the mother cells. But nevertheless, you could have a situation where one of these nodes controls everything in the tree that happens afterwards. There are subsequent divisions in the tree, or subsequent levels. And so in that sense, you could have a heterarchy. So this is all, the hierarchy here is all based on ranking. We rank things according into biology to their age, in organizations according to their sort of pecking order. And in biology, you have pecking orders too, just not in this example. So I wanted to make that point. And I think we're talking about hierarchies and we're talking about pecking orders, and this is going to lead us to networks. And so in this case, this heterarchy also forms a network, but it's a very different type of network. And so Herbert Simon's statement about a hierarchy being a rank order matrix with very low values, so we have to think about this now as a matrix with entries. And these entries, of course, then are going to be, we're going to have to put numbers in here. And so now a lot of these numbers are going to be close to 0, and some are going to be higher. And that gives us our hierarchy. But we also can get heterarchy out of this, because these values can change, and some of them can switch from near 0 to higher levels, higher values. So this is putting this all into this sort of matrix context. Okay, so now we talk about universality. And universality is interesting because, we talk about cellular automata in this group, and cellular automata are a great example of something that exhibits universality. Universality means that it applies across all different types of contexts, the different types of systems, whether they be biological, like developmental systems, or evolutionary systems, or human organizations. Those are all basically going to yield the same results or the same types of patterns. And so this is one of the basic aspects of networks science, is that networks like technical networks and all sorts of biological networks, social networks should all yield the same types of patterns. And so this is where he gets into some of this stuff about, you go back to Kant, who said whether free will exists or not, human actions, like every other natural event, are determined by universal laws. And so this kind of goes on talking about some of this. Perhaps the most well-known examples are Zipf's law, which is a variant of the power law. But Zipf's law, and this has been found in human languages and across other types of systems, cities, the size of cities, the distribution in a country or around the world, it conforms to Zipf's law. Zipf's law is basically the frequency of the kth most common word, and they're using this context of a dictionary of words, and this is where Zipf's law originated. The frequency of the kth most common word is inversely proportional to k, having an exponent of -1. So this is Zipf's law. And then Pareto's principle of wealth distribution, which is the richest 20% of the people have 80% of the wealth. And you can have this sort of inverse relationship between individuals and resources. And so you can see this in biology as well. We see this actually in networks, and we're going to get into how networks, when we talk about network control, certain nodes will have a large share of connections versus other nodes in the network, and those nodes are called hubs. And those hubs serve as the basis for hierarchy in networks. So this is all kind of related. There are different ways that people have formulated this over time. It's very interesting, but it all points to this universality. So in the physical theory of critical phenomena, the meaning of universality came to change in the 1960s and 70s. Critical exponents describing the phase transitions seem to fall into a few so-called universality classes. Describing how systems change with length scale, the renormalization group theory led to a theoretical foundation for the scarcity of critical exponents. Moreover, it motivated statistical physicists to search for universal laws outside of physical systems. It became a prominent example of how universality can appear from emergence. And that's another defining feature of complex systems. So we can look at universality and ranking dynamics. And this is where we look at some of this physics-flavored complexity science. We use this to sort of get at this question. So I think they have, this illustration here in figure 1. This is dominance relations within a group of leghorn hens. So this shows ranking and how this changes over time. So this ranking is obtained by a simple greedy relabeling, not necessarily the one minimizing them or discordant pairs. With respect to the ranking is so-called minimum violation ranking. So you can see that these rankings are sort of linear, but sort of nonlinear if you transform them. Okay, so there are 2 papers here, Bloom et al. And Iniguez et al. And they both use this physical way of doing this. This is from physics. There are probably other ways to do this, but people haven't really explored those because the physicists sort of have the monopoly on some of these tools and ideas. So these 2 papers, they build a microscope model of list dynamics that differ from the former. So these models are the same, but they're different in 2 aspects. So Iniguez is the first paper. They study open lists where, like many real-world rankings, only the top elements are explicitly ranked. So in other words, only the top parts of the list are ranked. The rest are free to move around, and we don't really care about those. Second, they do not assume the ranking is based on a variable associated with the items. So you don't have like a variable called rank. The ranking is just inherent in the model. The model of Iniguez consists of only replacement and displacement. So these are where items can move within the list and offset the others, or an out-of-list items can substitute an element. The universality in their model refers to a non-trivial consistency relation that the rates of replacement and displacement must obey, which they further corroborate with empirical data. So details of the displacement dynamics do not matter. So this is where things change rank over time, and the relation holds whether the jumps in the list follow a fat-tailed distribution or not. So we're talking about these distributions that are long-tailed. So again, I mean, I've talked about this in other meetings, but we have what is essentially this power law, and I'm just going to draw it in a way that's sort of linear log or something like that, where you have this function, and I like to draw it this way because it kind of drives home what the distinction is. This is the upper part of the rank here in a rank order. This is rank order, and this is like magnitude or something. Right? And then so this is the higher part of the rank, where you have like the most kind of the highest ranking things, and then down here you have the lower parts of the rank, which may or may not be ordered. You may or may not pay attention to the order here, but this part of the rank is most important, and this is where you see the most variation in terms of magnitude. This is where you have the most connections, where you have the most power, you have the most wealth, or whatever. And so this is the part that matters. This other part can be, ordered in different ways, but it's not so much important because, as you can see, it doesn't really contribute that much to our understanding of the variation in the rank, or why things at the top ranking things are sort of the most salient in terms of our understanding. Now, there are things that can move around in the rank, so things from this category can move up to this category. And so we can, have to account for that in some way, and that's what they talk about in the paper here. So there are different ways of doing this, different methods. And so we end up with this fat tail, which is down here. And this is basically tells us like the intensity of the hierarchy, intensity of the inequality, or whatever it is. This being larger, meaning that the rank is less sort of strict. And, maybe in hierarchical systems, this part of the distribution is very large. In fact, there might not be a fat tail at all. It might be a very thin tail in a heterarchical system. So if we go to a heterarchical system, we might have a graph like this. And again, rank order being relative because rank order can vary over time and things can change their rank. Something like this. You have this large zone of things that are high ranked. And of course, because they're always changing rank and there's a good chance that they'll all be like sort of the number one thing or the main hub or whatever, they would be in this area. So this is heterarchy. And this is, this is the difference between hierarchies, heterarchies, and all these universal statements that we can make, or universal things that we can find. So discovering universal relations of simple observables and list dynamics have more practical benefits than the lofty goals of Kant and others. They give us precise language to describe a phenomenon. We know that this replacement rate is sufficient to explain the state of ranking dynamics. Thanks to Bloom, we can describe an item in ranking just by the phase to which it belongs. So these graphs that I showed you here are all, they're all kind of speak to this kind of list, this list ordering and some of these things like that. So this is a very important topic in looking at these hierarchies. And when we just look at like one of these hairball networks, we don't necessarily get the sense that these are the underlying universalities. So, since rankings are rare, so should practical applications of the theory of ranking dynamics be. While it is hard to speculate about the exact form of such applications, highly predictable persistent patterns, the statistical laws discussed above, would be valuable as foundations. This reference here, number 5, also opens several new research direction avenues. So, first of all, we'll go through them here. The role of memory in real-world list dynamics seems to be uncharted territory. So memory would be if I had a list, and that list had some sort of memory. So it's kind of like a Markov process, but in list form. Okay. So your list is here. 1, 2, 3, 4, 5. And you get reordering. But this reordering doesn't occur at random necessarily. There's a memory. So one is, we remember that 1 is 1. And if it gets reordered, there's it's more likely that things will go to the top. So it's more likely that this will reemerge to the top more often. And so there's your memory aspect. And so that's one way to do this. And of course, I mentioned this is a Markov process because this could act as a Markov process, an ordered Markov process, or it could just act as something that's biased by having a memory. So a memory, not a memoryless Markov process. So yeah, this is in stark contrast to the memoryless models of these references. Second, because of their even more fundamental role for human organization, it would be interesting to study the dynamics of hierarchies. The big caveat, of course, being that there's no universally accepted definition of hierarchy, which we've talked about, and of course we can contrast it with heterarchy. So to fulfill the promises of complex system science, we need to keep discovering these fundamental relations of simple phenomena such as ranking dynamics, and they form the basis for more complex systems. I hope you enjoyed that overview. Hope you learned something. All right. Well, have a good week. And again, if people are interested in the Huggingface models or the GitHub repository, please participate. You can put a GitHub issue up, or you can issue a pull request, or you can join our Slack and ask questions there. All right. Thanks. Have a good week.
Bye.
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 Bye.